Have you ever wondered how fascinating our genetic makeup is? Take for example in women, the fact that their mother contributes to one X chromosome while their father contributes to another X chromosome. This means that women have double X chromosomes. Aren't there too many X chromosomes? Women would have double the amount of products of the X chromosomes, right? Well, surprisingly, they don't. It's quite intriguing, isn't it? So why is it that women typically have one pair of X chromosomes, but they do not produce double the amount of X-linked gene products? To find out more about this, let's look at the role of DNA methylation in X-chromosome inactivation. We are Juanqing, Matthew, Judah, Olivia, and Trixie from Group 1. Let's explore this together and uncover the wonders of genetics.
X-chromosome inactivation, or XCI, is a really cool mechanism that helps even out the gene expression level between male and female cells during the early development in female embryos. Basically, one of the two chromosomes in every cell become highly condensed and undergo inactivation or silence. Well, except in the X cells. What happened to the inactivated chromosome is that it will condense and thus form something we call the bar body. It's a really remarkable and complex of how our cells develop.
And why is it so important?
Again, women have two chromosomes, while men have only one X and one Y chromosome. Without this amazing X chromosome inactivations, women would generate double amount of the gene products. And of course, this would disrupt the normal developments and functions. Now, you're probably wondering how this mechanism works. So, this occurs in the X inactivation center, or XIC. Normally, X chromosome inactivations start from the synthesis of the X inactivation specific transcript, or ccRNA. ccRNA will bind and act as a scaffold for chromatin modifying factors. Usually, they are proteins. This means that this RNA will transport these factors including DNMT3A, G9A, PRC1, MLL, WDR5, and LSD1-CoRAST to the side of the chromosome. Together, these proteins will condense the chromosome, thus forming the so-called bar body.
Then what actually is DNA methylation?
As previously mentioned, one of the proteins that sends RNA recruits to the chromosome is DNMT3A. DNMT3A is a DNA methyltransferase, which is an enzyme that adds a methyl group to the fifth carbon of the cytosine DNA base, and this process is called DNA methylation. It is one of the markers of XCI, usually one of the final steps of XCI epigenetic processing. This process occurs during early female development, where upon initiation of the XCI, cis recruits methyltransferase to methylate the gene in the activated X chromosome.
How does DNA methylation assist in the inactivation of the X chromosome?
Well, DNA methylation helps to maintain the silent state by further preventing the binding of transcriptional machinery. It acts as a steric hindrance, blocking the binding site for proteins such as polymerase. Other than acting as steric hindrance, DNA methylation can recruit DNA methyl binding proteins that can recruit transcriptional precursor, which help to further silence the genes in the inactivated X chromosome.
But where does DNA methylation occur?
DNA methylation is established from X chromosome inactivation at the promoters of genes on the inactive X chromosome after several steps of epigenetic marking during cell differentiation. CpG islands, typically found at the 5' end of genes, are reported to be heavily methylated on the inactivated X chromosome in somatic cells.
Something interesting is that some genes in the XCI have the ability to escape. This means that, although the X chromosome is inactivated, some regions still have transcriptionally active genes. This is due to hypomethylation, or decreased methylation of the region. Research shows that genes that escape XCI will exhibit low DNA methylation in women, with both the inactive X chromosome, or XI, and active X chromosome, or XA, remaining unmethylated. In contrast, genes subject to XCI will show intermediate DNA methylation, with the XI methylated and the XA unmethylated. Some genes located near the transcription start sites, or TSS, on the XCI are known to escape XCI due to hypomethylation. Using methylation analysis, an XI DNA methylation level shows that the TSS is always subjected to XCI. However, DNA methylation level of under 15% shows that the TSS tends to escape from the XCI. Thus, understanding the DNA methylation landscape, which reflects CpG density and functional chromatin state, is important to understand the patterns of methylation associated with the XCI and XA.
Now that we are familiar with the role of DNA methylation in X chromosome inactivation, do you think that its application might be useful in the clinical setting? such as predicting disease, perhaps. Well, it is intriguing to consider its potential utility in predicting XCI-related disease. There are several X-linked diseases associated with the skewing of X-chromosome inactivation, such as hemophilia, both factor VIII and factor IX, and ornithine transcarbamylase deficiency. In these instances, heterozygous individuals experience varying degrees of severity of the disease, depending purely on how many of the involved cells are expressing the pathogenic allele over the functional one. On the other hand, dysregulation of XCI is a common hallmark of several types of cancer. In high-grade ovarian serous adenocarcinoma, reactivation of XI and overexpression of genes from XA can lead to increased expression of oncogenes, while in breast cancer, excessive exist silencing of BRCA1 prevents normal regulation of cancer cell division. With further research, we might uncover the role of DNA methylation in these processes, and if they can be used as a prognostic tool in clinical settings. We've known that DNA methylation plays an important role in the confirmatory step for XCI. Therefore, its patterns may provide insights into mapping the inactivation of X-linked genes and the assaying for disorders and conditions associated with skewed or aberrant XCI patterns.
Well, that's it from our group. We hope that this could give you an insight into DNA methylation and X chromosome inactivation in more depth.